New Year to all of you joining us tonight for our first webinar for 2021. Let's hope that this year coming would be a better year for all of us, for beekeeping, honey, and generally all around. A lot of you will know Professor Grace McCormack, who's kindly providing us with loads of new facts and information here tonight. And you will have read her profile there. She's professor, as I said, in NUIG, professor of zoology, and her interests lie in evolutionary biology and particularly in using molecular data to understand how organisms are related to each other and the impacts this may have on conservation and on the evolution of organisms and their traits. The interaction between animals and their parasites, pathogens, over evolutionary time is also of interest to her, as is the use of molecular information in applied science, such as biodiscovery, marine sponges, and apiculture. Grace has been keeping bees for about 7 years and currently manages 15 colonies. She became interested in free-living colonies in 2015 and has been investigating their survival, diversity, and distribution since then. She's interested both in conservation of Apis mellifera and resilience of untreated bees to varroa mice and other challenges introduced by humans. This talk tonight discusses the origins and diversity of the types of honeybees in Ireland. In the background, we have Stephen Gallagher, who's our technical guru, and he's kindly encouraged us all to grasp this technology in these times. So look, enjoy Grace's talk tonight. I've heard some of it before, and I know you'll be delighted and you'll be kept interested in all she has to say. Thank you.
Okay, thanks a million, guys, for inviting me tonight. I think these webinars are a great idea because I do think, well, beekeepers are isolated due to COVID, and I think these online meetings are great. I think communication needs to be improved and extended more than ever in these days with all the data that's coming out on pollinators and beekeepers that needs to be communicated to beekeepers and vice versa. This communication between science and beekeeping is really important. I just want to say first, this is collaborative work. Work I'm presenting. I've had the pleasure of working with lots of different people. On the left there is a team from NUIG who helped me beekeeping, and some of the students and researchers. And then some of the molecular work has been done in collaboration with these two ladies in Portugal. My PhD student went to visit them and we've published a couple of papers together. And then the team in LIT were involved with us collaboratively producing that paper in 2018 and our one this year on the wild bees. And Gabrielle Soland is from the company that has generated, well, through her PhD and stuff, she generated the microsatellite panel that has been used a lot, and so I work a lot with Gabrielle. But actually, even more than all of that, this huge amount of thanks due to the beekeepers and members of the public that have really gotten involved with not only kind of providing samples, but actually all of the research questions I'm asking have come from beekeepers. That's how I got involved in bee research, was through providing answers for beekeepers. And actually the why this talk came about was really, it was designed as a talk for the Tribes Association. I'm a member of Tribes, and during the summer I was trying to prevent hybridization. Some of my colonies and some of my queens were getting mated with a mix of drones, and I just asked the group, you know, what kind of bees they had, and did they know an area of Galway that was pretty good for native bees for true breeding. And I was surprised at how many people came back saying they didn't know what kind of bees. They didn't even really understand my question. So I think that's why I wrote this talk. And there's a lot of misinformation out there about what's what. And the other thing is, what I realized through my research since I started was actually we have two different bees we're talking about here. And beekeepers sometimes, you know, I guess we're focused on the managed bee cohort, but actually in the first and foremost, honeybees in Ireland were wild a wild species in this country. And while beekeepers are often touted as the custodians of bees, and while I agree with that to some degree, beekeepers are also bring much stress and pressures on bees. And while some beekeepers don't care about that, perhaps a lot of beekeepers do. And I just think it's important to keep in the back of our minds, whatever we do. This bee is a wild species. It sometimes in some of the literature is touted as a domesticated species, but this species, we can't control its breeding. We don't control its breeding here in Ireland to any great extent at all. It's a free mating species, and also we don't really control its forage. So it's not really a domesticated species, and we do have wild honeybees in Ireland. And so I think this is important for beekeepers to be aware of, and it impacts on your beekeeping, and it also impacts on the bee itself. And so I was asked to become involved in bee research, as I said, to provide scientific evidence for beekeepers. One was actually providing scientific evidence about the efficacy of a food supplement that was being developed. That's what got me started. And then also to look at a breeding program for varroa resistance, and also actually to find out what kind of bees we had in Ireland. And when I started, I was surprised really at the reading. I was surprised at the lack of research. I'm trying to minimize that. Okay. And basically when I came, it was thought that a lot of people I was hearing were saying there was no native bees in the country and no wild bees in Ireland anymore. And some of this, the two quotes above are fairly from old data. So on the left, the original IBA, when it first started, the Irish Beekeeping Association back in the 20th century, very early. In 1923, some of the first minutes of the meetings and letters reported that this country was swept clean of bees. And this corroborates really what Brother Adam, so this was the brother from Buckfast Abbey in the UK, who was an avid beekeeper, and he basically also said that native bees in the UK were completely wiped out by the Isle of Wight disease. And at that time, that was thought to be the case, and this was perpetrated for quite a long time. And Brother Adam at that time then developed the Buckfast bee in order to really, for very good intentions, in order to make a resilient bee that would not be wiped out by something like the Isle of Wight disease. But you know what, the reality then has become clear as more and more people started to look at what happened, was that the native bee was not wiped out by the Isle of Wight disease, and it wasn't just a single factor that caused that population crash anyway in the first place. There were a number of different things that occurred at that time to cause, yes, there was a population decline in bees due to the war. Lots of people were busy with war interests and not spending time beekeeping, and there was certainly disease as well. And so it's thought now, subsequent to that, Norman Carrick has done quite a lot of work on this to suggest that Brother Adam wasn't quite right. And although the reason for him developing Buckfast was very laudable, he was incorrect in his original assumptions. And unfortunately, because he's a brother in some contexts, and the Buckfast has become very popular. And so this can be an issue. And then there was a lot of work saying that there's no feral bees colonies left because of varroa. So in 1998-99, varroa came to Ireland and caused another huge population crash of bees in Ireland. And for sure, there was a huge demise of wild bees and as well as managed bees. But the National Parks and Wildlife Service, who really are the government agency responsible for looking after what's kind of our unique biodiversity and protecting the biodiversity we have, really don't engage with bees at all, honeybees at all, because they're described as a domestic species, so they're under the remit of perhaps of Department of Agriculture, Food and Marine, and also because they thought that there were no native bees. And I was surprised at the number of different sources that stated this, as if this was true, from biotechnologists, conservation biologists, beekeepers, members of the public. And what I couldn't find really was evidence for any of this. And then you come across statements like the native Irish Buckfast bee. And so there's a lot of statements and opinions given in what we call the grey literature online and magazines and reports and stuff like that, but very little published research. So that's what kind of I started doing, was looking at what we had. And I'm just going to talk a little bit, I'm going to talk fairly quickly because I have a lot of slides to get through. So hopefully, at least if I'm talking quickly, I'll be talking clearly. And I'm happy to take any questions, of course, afterwards and explain methods or whatever is necessary. So first, just the background of kind of the approach that I've been using. And this is using Darwinian evolution at its core. So the idea of natural selection and the survival of the fittest, but also attached to this is what we call the modern synthesis. So at the time of Darwin, when he wrote The Origin of Species, and he talked about natural selection as being a driver of change in populations, he didn't know anything about genetics at the time, because the genetics, the discovery of Mendelian genetics with Gregor Mendel, a monk working on his peas, wasn't known. And this is really unfortunate. But later on, when the structure of DNA became evident, and the work of Gregor Mendel, there was a lot of work putting together this kind of genetic approach with Darwinian evolution. And this is what's called the modern synthesis. And we can look at it really simply, that evolution is just change over time, but it's change really in inherited traits. So it's not change in traits that can be changed over an organism's lifestyle, like, you know, building body mass and things like that. These are traits that are inherited. And this is reflected, just the power of evolution in causing this change is just shown here by the picture. So I have an ancestral apis here, which obviously there's no picture of because it doesn't exist now. We don't have a picture. But over time, changes in these inherited traits gave rise to these multiple species that you see all around. And some of these pictures were taken by John Breen, amazing pictures of these species. He gave me instructions of things to say, but I don't have time about their morphology and where he found them. But so you see that from an ancestral species, multiple species have arisen of different species that are related to each other. So they're all in the genus Apis, and that is, so they all have traits that can be identified to place them in a single group, but they're also distinct amongst themselves. So they can't breed with each other. They're separate species, so they only breed with their own kind. And of course, the one we're interested in is the one at the top, Apis mellifera. So these evolution causes these changes over time, that allows us then changes in morphology that we can see. And these traits are inherited via the DNA. And DNA, you know, it's just no harm to kind of have a think about it, because actually what I'm studying is this DNA molecule that represents then the change, the evolution in the organism itself, as a proxy for what we would measure in the animal itself. So here we have a picture of a cell, and inside the cell you've got the different organelles that carry out the functions of the cell, and in the middle is the nucleus, and the nucleus contains all of the chromosomes. So if we take the nucleus and crack it open, we see that it's filled with these chromosomes, and these are basically tightly coiled packs of DNA. And they're packed together with proteins to help keep them safe and stable, because the DNA is this chemical molecule that codes for all of the development and the functions of the organism. So if you stretch out these chromosomes, you see the standard double helical structure of DNA. And the beauty of DNA as a molecule for allowing inheritance is that, firstly, when the cell divides, the chromosomes divide. And when the cells in the reproductive organs divide to form gametes, then the chromosomes separate out into egg and sperm, depending on obviously whether you're male or female. And so one copy of a chromosome will go in each sac cell, and then later on when they reproduce, the egg from one and the sperm from another, that DNA that's contained, that represents those lineages of the male and female, reunite then in the fertilized egg. So if you look at the DNA structure then down the bottom, the DNA structure is comprised of, in the middle, you've got these 4 chemical types: A for adenine, T for thymine, G for guanine, and C for cytosine. And these represent the 4 bases you often hear about and see in alignments, A, T, C, and G, which are what we study when we study DNA. And those are arranged in a very particular way along the DNA because they contain the information. And you see that you have 2 copies that are joined together by bonds. So A is bonded to T and G is bonded to C. That's the normal way of things. And they are kind of held together by the sugar phosphate backbone, which is just the backbone, or the sides of the ladder. And basically when the cell divides, or these chromosomes divide, basically the bonds are broken between the A and T and the G and C all the way down the length of the DNA, and that double helical structure separates. And the information on one strand is used by enzymes in the cell to make the other strand of DNA. And what happens when that DNA gets replicated is that sometimes mistakes happen. Now, higher organisms, we all have, oh, I didn't mean to go there, but we all have these enzymes that fix the DNA, and sometimes they're not perfect. So errors happen in the DNA when the DNA has been replicated, either sometimes when cells have been replicated, and certainly in the formation of the egg and sperm. And then these get perpetuated or inherited to the next generation. So with this idea, so the picture on the top right is of a cell with the chromosomes, and these chromosomes contain DNA. And so if you stretch out that DNA and look at it, we see that the DNA is packaged into different sections. And each of those sections often codes for a gene. So the DNA contains the genes of the organism, and the genes contain the instructions for making all of the proteins and so on in the body. And all of this DNA together comprises the genotype. So when we talk about something's genotype, we're talking about the type of genetic DNA that's present, or genetic information that's present in that individual, in all of the cell. Now, the cell contains a nuclei, and I'll talk about mitochondrial DNA later, which is present in some of the little organelles. But basically, fundamentally, the genotype of an organism represents what is the type of DNA that's present. And the DNA, then, as I said, along the length of it is coded into genes, and each gene generally, I'm oversimplifying to a certain degree, makes a protein. And the protein then is what makes the bulk of the organism. So the morphology, the muscles, the hair, the eye color, and all of that, and the enzymes that drive metabolism. So when you get changes in DNA, you sometimes get changes in the protein. And we can look at that. We can compare the changes and the differences in DNA, which would mean that different individuals might have different genotypes. And the bottom, we have something called a phenotype, which is the physical appearance of the individual. So this is how, I guess, the genetic information is manifested in as the morphology and behavior and metabolism of the organism. So at the bottom, we just have 3 different species, and they look different. So those differences in what we can see represents the phenotype. So these are observed and measurable traits that are as a result of the DNA. And in between those things, the environment is operating. So the genes are being expressed in different environments, and therefore that will have a different impact on the organism. And it can change the phenotype, and this is where the environment, this interaction between the environment and the genotype, is where we get natural selection. So we look at some schematics like this to describe that genetic variation. So basically, if we look at the diagram here on the right hand side, and you look at the top, represents the common ancestor, where those branches join together at the very top is where we would represent the common ancestor of all those 4 contemporary populations at the bottom, A, B, C, and D. And that original ancestor divides into 2, there might be 2 populations that are slightly different, and that they, as time goes by, if they're separated, then the more time that goes by, the more DNA change will happen along those lineages, along those different branches. So you get greater divergence over time in populations that are separated. And then the longer the time goes by, and if these populations, as say, are isolated, i.e. they no longer reproduce with each other, they don't swap genes anymore, then the more protein divergence will take place, because DNA codes for protein. And then the longer those populations remain apart, then thus proteins start to come into play, and you get changes in the anatomy and the behavior and the physiology of these populations. And so at that point, sometimes reproductive isolation can be cemented, where you get changes in mating songs and mating patterns and even mating structures. And so we can use DNA to reconstruct diagrams like that to show how populations are related to each other over time, and how much of this divergence may have taken place. So natural selection, the fundamental kind of idea behind this is that in any population of a species, individuals vary. And this is really important, actually. So the individuals that are better suited to a particular environment are more likely to survive and reproduce. That just makes common sense, that quite often resources are limited in any place or at any time. And quite often if a species is well adjusted to an area, it will often produce more individuals that environment can take. And therefore you can have variation in fitness. Provided this variation is genetically controlled, the reproducing individuals, then the best fit ones, will pass these inherited characteristics to their next generation. And then over time, you see a gradual change in the population, because those individuals that are fitter have particular traits. And if they become more common in the population, the traits that are present in the individuals that are less fit will start to disappear. And this is called adaptation. And this is represented here in those pictures, whereby you do see this natural selection operating in honeybee populations, whereby you see a very distinct difference between subspecies, populations of this species living in the south, like Ligustica at the bottom. I must have this on a timer because it's jumping without me saying it. So I'm sorry if I'm jumping back and forth. I was recording this this lecture to make available, and so there must be some residual timing in here. So as I said, yeah, so these population here in the south is, say, Ligustica, and then the ones that are adapted to the northern Europe, the cold, damp, wet climates, are darker, and they have other supposed adaptations as well to make them more suitable to those conditions. But we can see these changes ourselves with our naked eye. So this variety that's in a population is really, really important, and anything that really undermines or removes this natural variation is really to be criticized or to be avoided as far as possible. So this is the classical issue that happens with populations that are threatened with extinction and endangerment, is when the populations get very small, they lose this genetic variation and they lose the power of adaptation or natural selection. And what this variation gives us is a natural adaptive capacity. One hand, it gives us this geographical capacity, which honeybees have really excelled at. So they have a huge variation across the geographical range in which they exist. And therefore, it means that if you have a change in geography, or sorry, a change in environmental conditions in one location, if there's variation in the population, it allows the organism to adapt. So it would be very important for us, for example, that some honeybees have an adaptation for climate change and global warming. Other things like competitive challenges, you want natural variation to be there so that that allows this survival of the fittest to have a very good edge, and so that you really have organisms that are very, very well adapted to current conditions. And then pathogen challenges also, the more variation you have in a population, the more likelihood there is that there will be some individuals in a population that can withstand a new pathogen or an alteration in a pathogen. So the classic example I use with students is with cheetahs, for example. The population is so small that their immune systems have very little genetic variability. And when there was an outbreak of feline peritonitis in the late 80s, cheetahs lost like 90% of cheetahs because they had no resistance to that pathogen, whereas domestic cats were highly variable and it hardly knocked a stir out of them. And this is really important for, we would hope, and I think that there will have been some population, some bees in Ireland that were able to withstand varroa, and they have survived. And these are the bees that now beekeepers are able to use to basically avoid having to use treatments, and that this capacity exists in these populations to allow us to do that. So, and the other thing is, of course, is that as well as natural selection, natural variability in a population allows us to artificially select a species for traits that we want. And you can see that very clearly in cows and dogs and horses and in bees, actually, in terms of artificial selection. We don't do it very much in bees in Ireland, but they certainly do it in other places. So this idea then is really important, that environmental factors vary from place to place. So like and basically an adaptation in one environment might be very, very useless or detrimental to the organism in another environment, and vice versa. So we have two pictures here. The top one is outside my house in November, where you have this dank, dreich kind of a day. And in Northern Europe, the bees have adapted to this and the wind and the rain and the misery anyway, and they have developed in order to be able to fly on damp days and windy days. It's thought that the black bee has got some much sturdier wing muscles to allow them to fly in these conditions. I've not checked that out yet. And here in the south, we have got southern climates, like a lovely Italian climate, and you've got bees adapted for that. So when you bring these yellow bees into Northern Europe, they don't do so well. The adaptations that many of them have would be fairly detrimental to survive a winter in Ireland. Now there's some talk that say Carnica bees from mountainous areas survive very well in Ireland, and it may be that the environments are very similar. So it's important to think about the environment in which your bees are and what bees are more suitable for where you are for beekeeping. So this has led to, in the honeybee, it has led to the evolution of a number of different subspecies. So these subspecies can still, in theory, interbreed with each other and provide viable offspring. So they're classed as the same species, but they're very, very distinct. And so they're called subspecies or races. So evolution is taking place. And the idea is, I suppose, that over time, if they were left to their own devices, and these barriers that you see, like the Alps here, for example, mountain ranges and so on, are causing barriers for honeybees to cross. And if they were left to their own devices and these geographical isolation mechanisms, then you could conceive that they could eventually become new species. But of course, humans have come into onto the stage and removed some of these barriers before full species status has happened. So this slide just is talking about that speciation is really when you get the formation of a new species from an ancestral species. And we saw the ancestral Apis species has evolved into different species of bee that are shown here on this slide, including the honeybee. But this type of natural selection and geographical isolation is happening all the time within the honeybee populations. So when you get environmental separation of populations, so like, for example, this picture on the left is the Grand Canyon. Mountain ranges are also very common. So where the organism has a difficulty to cross. You get adaptation to environmental differences on either side of the geographical boundary. And over time, as the schematic I showed before shows, over time, the lineages accumulate more DNA substitutions, more differences in their protein, and then more differences in their behavior and their anatomy. And they go on to evolve different entities. If, for example, the barrier gets removed before full species status, before complete reproductive isolation happens, then you can get basically mixing happening all the time. In some cases, between some species, you get hybrid zones whereby where the two species meet, you still get some hybridization between closely related organisms. So this is what happens in evolution. So as this variation and divergence happens, the end result will be new species unless that boundary gets removed. And so if we look at Apis mellifera, the honeybee, this is a paper that came out last year, but there's a lot of different papers indicating the different numbers of entities or races or subspecies that have evolved within this species. So technically they can all still interbreed, but you can see there's a huge diversity. So 33 different subspecies across 5 major lineages of these bees. And you can see them spread out over a large geographical area and some very different geographical locations. And 14 is Apis mellifera, which probably has the largest distributional range of any of the subspecies. And these subspecies can be distinguished by morphology and DNA. So you know that better than I do. And these are two papers that I found really interesting because there are some kind of patterns that we really have to investigate with Apis mellifera that really will have big impact in terms of its management, I think, in places where other subspecies exist and are brought in and are liked by people and want to be held by people and so on. And this is the idea of really the extent of reproductive isolation of Apis mellifera and how we might kind of manage that. So the two papers I've come across, one is in Poland where you have A.M. carnica and A.M. mellifera coexisting in Poland. And actually they haven't completely hybridized into one entity there. So they're maintained as separate entities and they have, I think, a small hybrid zone. But these guys, what they did was they tested the queens of Apis mellifera mellifera and Apis mellifera carnica, and they artificially inseminated them with semen from both subspecies. So mellifera, they inseminated her with 50% sperm from mellifera and 50% sperm from carnica, and they did the same with the carnica queens. And then they looked at the offspring, and what they found was that with Apis mellifera mellifera, she bred true, and she only used the sperm from the mellifera drones. So there was no carnica drones sperm used, whereas the carnica queen didn't show that specificity, and 50% of her offspring were sired by mellifera drones and 50% by carnica drones. And I found that really interesting. And then the other study was done in Tasmania, and where they showed, this is with mellifera ligustica here. And what they found was in the mountainous areas in Tasmania, and you might think, you know, in environments maybe similar in some context to Ireland, you found that actually the ligustica drones were less fit. And so the queens in these areas bred true as well. And so because they didn't use the less fit sperm from the drones from ligustica. But down in the rest of Tasmania, where it's hot and dry and warm, where there might be environment more similar to what ligustica is used to, then the ligustica drones were very fit. So I do think the fitness of the drones and the environment in which they are plays a huge role in what the Apis mellifera mellifera queens will do. And I think this is really important to be thinking about in management of this situation in Ireland. And I'd like to do some work on that in the future. So for this section, really just to summarize, it's what I'd like to say is that mutations over time in the history of mellifera in Ireland, and since the origin of the honeybee to start with, mutations accrue over time. And as honeybees expand their range and they have moved and they have spread out from Africa and Asia into new environments and coming up further north, basically these substitutions have led to the formation of these different subspecies. We call them races, we can call unique variants, and each subspecies and local variant are very well adapted to the local environment. So as they moved out, they became adapted, and the fitter bees that were slightly different to the previous bees were what survived and reproduced, and is led to this pattern of subspecies throughout Europe and Africa. So what kind of bees do we have in Ireland then after all of this? And this slide just talks about where did Ireland's honeybees come from? And the first situation is that it is now assumed and accepted that bees reached Ireland naturally from Europe, probably across the land bridge from France to England and then to Ireland after the last ice age. So as the ice began to melt and as the ice retreated, there was a period of time by which the land was connected in the edge of the continental shelf. So France was connected by this green here to England, and then there was thought to be some connections between England and Ireland. And then the period of time, the sea level started to rise again, and Ireland became disconnected from England, and then eventually England became disconnected from France. And there's talk that as the oak trees and stuff extended up after the last ice age receded, then a lot of insects, including bees, came up north along with the trees. There is some suggestion to think that perhaps some fauna and flora arrived in Ireland through Iberica, so up from Spain. But I think the land bridge is probably the most likely scenario. Some people argue that there was no land bridge between Ireland and England, actually, so this is only modelled because, of course, nobody was there at the time. But the entomologists suggest that even if there wasn't a very big land bridge, that lots of insects made it over to Ireland naturally, and that honeybees would have done the same, that the distance was not that great. And then we see lots of reports in the old books and papers that there were bees brought over from England around 500, 600 AD on boats, and that bees were rare in Ireland at the time, but they were present. And that was a situation, I would say, for a long time. And then in the 1920s, we had this collapse of the population in Ireland and England. And then the government at the time, there was pressure to bring in more bees, and the beekeeping was really important and getting popular again. And so the government brought in a lot of Apis mellifera mellifera from the Netherlands, reports of these skeps coming in, and there's memories. Micheál McGillicuddy talks about, he remembers as a boy his uncle getting him bees from the Netherlands. And we also have imports of ligustica from that time, and then other imports as well, I'm sure, of buckfast and carnica in certain amounts. So there is this kind of phased kind of introduction of bees, and then in more recent times, of course, the importation of bees has snowballed. There's a lot more importation of different things, of Mellifera and other subspecies into Ireland. But it's clear that Apis mellifera is the only honeybee that is native to Ireland. And native there's a couple of definitions of what it means to be native. One is that it has got here under its own steam, and the consensus is that honeybees made it to Ireland under their own steam around that time when the Ice Age receded. And then secondly, if the animal has been here long enough to have woven its way into mythology and the history and the language of the location. And bees have also done that. They form part of our language. They're in the Book of Kells. They're in the bee laws, the Brehon laws, and so on. So bees have been here for a long time, and under both of those definitions, they are defined as native. And they have evolved here, as in the rest of Northern Ireland, to adapt to our climate. So they're dark in colour, as we said before, to absorb the heat in cool summers. They apparently have adaptations to wing and flight muscles to enable them to fly in the windy or wetter conditions. They are docile. Of course, every population is going to have variation, which you want, but we have to test this. But there is reports of that native bees are aggressive, and some people think perhaps that people underestimate the amount of carnica influence in that, and that when bees hybridize, that certainly the second generation, the first generation is very robust and very prolific and great at bringing in the old honey. But then by the second generation, because the DNA of carnica and mellifera are fairly distinct and have been separated for some time, that they're not quite compatible. And when you get to the second generation of hybrids, that you have issues. And aggression seems to be one of those issues, but it hasn't been proven experimentally or scientifically yet. The native bee is supposedly very, very well adapted in other ways, in terms of the colony size being small, long-lived, good honey production, pollen, propolis production, frugal with the stores. So when forage declines, then the queen will come off lay and reduce the brood, whereas other subspecies don't do that and they need to be fed. And there's some talk that native bees are resistant to disease. So these images I got from Owen McGillicuddy. So we have a native bee that's adapted, and then what else do we have in Ireland? And some of these bees have been here now, I guess, for 100 years. So a while, but not that long in an evolutionary context. So buckfast is probably a very common one that has been brought in, and partly due to the popularity of Buckfast Abbey and the religious connotation, and it has been quite, at least I've heard from some beekeepers, that this is one reason why they've got buckfast bees. But also buckfast bees are very well loved by their beekeepers, and they're nice bees to work with. And so they're quite common here. And we also have carnica, which I guess is a worry, because if beekeepers are using the colour of bees to determine subspecies status, and if they've got dark bees, there is a slight concern that we might be not detecting carnica as easily as we can detect the other two. And we have ligustica. I haven't found very much of this subspecies, but it is in Ireland. And so there are others as well being brought in small numbers, but not really reported. Apparently around 115 queens imported per year legally, but we know that many more are brought in illegally from different places, and this can be an issue for disease, amongst other things. So if we're looking at the kind of the proportions of these other things, these other types of bee that are present in Ireland, we don't really know how many. And there has been, I guess, quite a lot of animosity between beekeepers on this topic, and it'd be really nice to try and reduce that and get people working together. What I can see is that if we look at Facebook as any indicator, the Buckfast and Carnolian beekeepers have about 237 members, whereas the Irish beekeeping one has over 5,000 members. So there's a huge amount more Mellifera present in the country. And also if we think about the fact that Mellifera seems to have some reproductive isolation, I don't think it's reproductively isolated against buckfast. However, I think this is a problem, but let's see, I think there's something here to work with. So the numbers are not very high. If we look at the map again of the distribution of the different subspecies, that's the source of where I got this paper. You see that the M lineage, so Apis mellifera mellifera belongs to an M lineage. So it's a very distinct lineage from Carnica and Ligustica, which are just below it here, just south of it, but are part of a C lineage. So they're fairly distinct. Within the M lineage, we have Iberiensis and Mellifera. So they would be more closely related to each other than either of them would be to Carnica or Ligustica. So Carnica and Ligustica are fairly distinct from Mellifera. And I just want to address a couple of things here, because I think misinformation, and we've just got rid of Donald Trump, so fake news and misinformation, we know what the damage it can do. So it would be really nice to just be all on the same hymn sheet, or singing off the same hymn sheet anyway, and know what's going on and what's right and what's not. So websites where you see "Welcome to Native Irish Buckfast Queens" is just, I have a big yellow X here, because that is just so incorrect and so misleading, and it's really bad because you've got beginners coming in who don't know what's what. You've got experienced beekeepers who don't know what's what, and when they see Native Irish Buckfast, they think, "Way, I'm going to get that." But Buckfast is not native to Ireland. So Irish buckfast queens, fine if they're being bred in Ireland and they really are being bred here, but they're not native. So Brother Adam created buckfast after the apparent demise, which we know is not quite true, of Apis mellifera mellifera. And originally he mixed Mellifera mellifera and Ligustica, but he later added other subspecies to this. So you would expect a fairly mixed hybrid bee to be when you hear of buckfast. And yet we haven't sequenced very many buckfast because I think this animosity going on. We really don't understand what's happening with how well Carnica is doing or buckfast in Ireland, and it would be really good to know so we can just all get along a bit better. So we have Apis mellifera mellifera by Apis mellifera carnica is what we tend to see the buckfast as being. Ligustica does hardly rear its head at all in what we've seen in buckfast. So I've only sequenced a few from Mayo, and these are clearly, well, when we do the analysis, I'll talk about this kind of analysis in a minute, but when we do this analysis, we see that it's really C lineage and Mellifera lineage that comes out for buckfast. But we don't know what buckfast is in Ireland right now. Other kind of misinformed quotes that really get up my nose are this one, for example, in time where honeybees are an endangered species on a global scale, should we be still insisting on the pedigree standards, and is it ethically responsible to kill a healthy and reproductive queen? Honeybees are not endangered as a species. They're everywhere. We have our native bees endangered across its range in Europe. Other bees like the Malta bee and other subspecies of honeybee are critically endangered and maybe even extinct. But honeybees as a species there's a lot of them. Honeybees have had a colony collapse because of varroa and so on, but they're not endangered. And also, people don't kill a vigorous queen because it's aesthetically desirable. It's not that people like black queens, it's because black queens represent a native bee. And so this kind of superficial kind of argumentative stance really doesn't make any sense. At the end of the day, the goal of any breeder is to first and foremost produce bees that are docile, healthy and productive. That's fine. But I just want to come back to the point that there's hardly any, well, there's some breeding going on in Ireland, but most beekeepers are hobbyists and like the bees. And basically, yes, a role of a breeder might be to improve their bees, but that's not the agenda for everybody. And again, to come back to say the bees are a wild species here as well as a partially domesticated species. So we've had a lot of impacts on Irish bees, the Isle of Wight disease, that first population crash. We've had a loss of habitat. Hybridization is becoming a problem. How much of a problem it will be, I'm not sure. We need to do a lot more work. Introduced pests and diseases, we know the impact that varroa has had, and we're worried about foul brood and also, of course, the role of pesticides. So honeybees are having a hard time. And the native bee, Apis mellifera mellifera, has had a huge massive decline in the population in recent years. The picture on the left shows the natural distribution of this subspecies, and the one on the right shows what the proposed distribution was in 2009. So it was assumed to be extinct throughout most of its range. Wild bees were extinct across Europe. The 2014 red list of honeybees stated the input for honeybees has been data deficient because none of the review group, who were all honeybee experts, knew of any wild populations of the honeybee across anywhere in Europe. And this included Ireland, apart from the Galtee, Micheál McGillicuddy was known for producing and protecting native bees, and he got a lot of stick for that. So does Apis mellifera exist anymore in Ireland? This paper from 2005 shows, this is just a distance matrix. The numbers just shows the relative distances between the different populations. And now there's a raised hand. Hang on now. Should I stop sharing or anything? I just see a raised hand. Will I just keep going? Am I okay for now?
Yeah, keep going.
Yeah, so basically they show that paper in 2005, they just included 48 bees from around the Galtee area. And they show that they were more similar to UK, but there were very, very few bees studied at the time. And then our paper in 2018, and we had the paper on the wild bees published just the end of last year, shows actually a very widespread population of pure Apis mellifera exists in Ireland. So we actually probably are one of the major strongholds in Europe of this subspecies that's in decline and has actually gone extinct in many places of its original range. So we have something unique here in Ireland. So I have to really hurry on, I'd say, because you're probably all going, it's 10 to. So we extracted DNA from the legs of bees, and we actually processed both the chromosomal DNA, the DNA that's in the chromosomes, and also the DNA in this mitochondrial. The mitochondrial produces the energy of a cell, and the organelle has resulted from, it used to be a bacterium that came into that cell, and it has its own DNA, and it has a lot of traits that are very informative of population genetics. And Lionel Garnery's lab characterized this region of the mitochondrial DNA between these two genes that was variable in bees. And the beauty of this is that it just represents the female line, so it tells you what lineage the queen was. But there's no influx from the drone, so you can't really detect, you cannot absolutely at all detect hybrids or anything with this method. And when we look at the mitochondrial DNA, we find actually that a lot, over 50% really of the bees, more of the managed bees and fewer of the wild bees, have a Dutch variant. So this really picks up the relationship between Ireland and Dutch bees. We also see some French bees in here, and one bee that's unique to, or identical to the bees in Colonsay, and represents swapping of bees between here and there. We have a lot of, quite a lot of unique mitochondrial variants as well, 29 novel Irish variants. So while we see variants that are present in Europe, we see also variants that are unique to Ireland so far. Now there's not a huge numbers, there's not thousands and thousands of bees in the databases for comparison. So actually the Irish situation, we've probably contributed as many sequences now to the database as were there before. So the other method we use is microsatellites, and these are DNA fingerprinting that have been used for like paternity testing in lots of different species, including humans, and for CSI and stuff like that. And so these are a particular region in the chromosomes that have these repeat regions. And what it is that they're small little regions of DNA like CTT here, CTT, that's repeated multiple times. And the number of times it's repeated differs from bee to bee. So this is a made up example, but in this B1 it has 7 repeats, in this bee it has 8 repeats, and in this bee it has 9 repeats. It's more likely to be longer than that. But that just shows you, and you basically, we look at a number of different satellite regions. So we looked at 12, and we look at it in lots and lots of bees. And when you analyze that data statistically, you get some very nice data back. And what we see, this is a graph of the mitochondrial data. And it's a program that basically looks at the data that comes back from the microsatellites. So every single bee has got a unique fingerprint from each of the 12 microsatellites we looked at. And it compares all of these bees from Ireland with, we got reference populations of bees from Gabrielle Salon's lab in Switzerland that represented Ligustica bees, Carnica bees, and other European populations from France and Norway, Switzerland. And what the program does is it takes the data from each bee and it assigns each bee to a cluster with a certain probability. Now I'm going to move slide for 1 second to show you an individual bee. So this is what we get back, say, it's hard to explain this when you know, somebody who's never done this analysis. This is basically a graph, a histogram, a very, very, very big histogram of nearly 1,000 bees. And each narrow little stripe represents one bee. And if we zoom into that, this is what it would look like for every bee. So every single column here is the result of a bee. And if we take here that red represents the C lineage bees, so Carnica or Ligustica, and green and blue represent Mellifera bees. Bee 51 here, the very first bee, belongs to the C lineage cluster with a probability of 1. And this gives you, this is basically that there's absolute certainty, like 100%, this bee can be assigned to lineage C with 100% certainty. If we shuffle over here to bee 64 here, 642, this bee can be assigned to, the red is the C lineage with 0.8, so I'm working the other way, and M lineage with 0.2-ish. It's not perfect. So this means that the program can't assign it with 100% certainty to either one of those clusters because it has some alleles, or some of the bits of the microsatellites are more similar to M than they are to C. And so this is what a hybrid would look like. And then if you look at 75, you see that it's roughly just under 0.8. The program says with a probability of 0.78 or something, I will assign this bee to M lineage. But there's a 20% chance or a probability of just over 0.2 that it belongs, that I'd have to put it with the C cluster. So this means that statistically the program has a difficulty plotting it with C or M, and therefore this is also a hybrid because it contains similarities to M and similarities to C. So I hope that makes sense. And the cutoff is about 0.9. So it's like a 90% confidence that the program can put your bee into one of those clusters. And the clusters, it defines those clusters based on the data, the microsatellite results that came back. So this program here, this picture represents many, many more bees. So it's just that the columns are much narrower, so they fit here. And so along the x-axis, you see the populations as we describe them. And the red and green represent the clusters that the program finds. So the program finds actually in this context that the majority of the data really separates C from M. So Ligustica and Carnica are both C lineage and they're together, and European Mellifera and Irish Mellifera are M lineage, and so they're together. And in the middle here you see, in the Irish Mellifera, you see some red, and these are bees where the program cannot with absolute certainty place them in Mellifera, so there is some evidence to suggest that there are some similarities to C. And so these are hybrids. These are actually 4 buckfast bees, 2 from one colony and 2 from the other. And then given, if we look at the 0.9% cutoff, then these bees are also hybrids, this one and this one and this one. So we've got very few hybrids. Now I want to show you this picture. I only generated this yesterday, the day before. I sent off another few hundred bees off to be for microsatellites. And just because I can't resist showing it to you, you're probably tired and bored already, but here we have again 1 and 2. The red are Carnica and Ligustica. The blue is European populations. And then the green, look, what it's finding here is that it's not coding all of this in blue. It's actually finding that in the Irish bees predominantly we have a new cluster. So we have genetic distinction of the Irish bees. So they show all this green cluster is present predominantly in the Irish bees only. There's some bees in the European populations that show some of these alleles. So alleles are just some of the variants, the microsatellite variants, genetic variants that we found. But most of them are in Ireland. So this shows us that not only are most of Irish bees that we sampled Apis mellifera, But actually we've got something very unique in Ireland here. We've got some, either they're very ancestral alleles from the old native bees, or it's new divergences. So we need to do a lot more analysis to figure that out. Other things very quickly to point out here is that I sequenced some UK bees from like Wales and Yorkshire, from beekeepers who are really trying to have black bees. And you see that they have a reasonably high proportion of Mellifera here, but a lot of hybridization. Well, the red is the hybridization, so this red here. Population 11 are the Dutch bees. And so you can see actually that the Dutch bees we process for microsatellites are more like the other European populations. They share probably more of the green cluster than other European populations do. So this shows this connection with Ireland, but I need to investigate that further as to what it means. And 12 are just some Iberiensis that we processed as well to have a better reference data set for the Irish bees. And again, you see that Iberiensis is in the M lineage and predominantly shows more similarity to the European populations, but there are also some of these green bits that is why I kind of think some of them might be old ancestral alleles. So this is not published yet, and I'm hoping that, I think from this I'm going to try and sequence more, process more bees from around the country independently and try and investigate this further, get a big, nice, big fat data set for publication. Just, I'm not going to talk about this very much, but just to say we also use the single nucleotide polymorphisms, and that show the exact same thing, a very high proportion of purity of AMM, both in wild, these contain quite a lot of wild bees as well. And that actually, I should show you here, 8 is the managed population and 9 is the wild population. And you see actually that 8 and 9 actually represent the same population. So feral, not feral, but wild, free living and managed bees in Ireland are the same population. This is a very quick study, and I'm showing it because I think you'd like to see it, and it's unpublished. It's from Jack Hassett's PhD thesis that I borrowed from the library. And basically you can see they did a study in collaboration with some of you guys, and where they looked at a Buckfast area in Kerry, where there are also Mellifera, to look to see whether there was, you know, I suppose the impact of each bee type on each other. There's some geographical kind of, there may be some geographical, what's the word I'm looking for, features here that keep these separate, but you've got two apiaries here with native bees, and then you've got these Buckfast beekeepers here. And basically what I just want to show you is that, you know, the test was really to see whether, the purity of these bees, to see whether they impacted on each other. And the 1 and 2 here are the native bees, and green here represents Apis mellifera, and you can see that the native bees in this area in Kerry were able to maintain very high levels of purity of Apis mellifera despite the presence of Buckfast in the area. And then the Buckfast bees were really surprised again, because they don't show very much ligustica. Blue is ligustica, red is carnica, and the green is mellifera. So what you're seeing in the Buckfast here is actually a more or less a carnica mellifera cross with Irish mellifera genes rather than anywhere else. So it seems to be that well, my hypothesis is that the mellifera, Irish mellifera, are infiltrating Buckfast and probably have a bigger impact on Buckfast than the other way around. That's not to say that Buckfast has had a huge, people bringing in Buckfast to an area where there's native bees has caused issues as well, because they do cause hybridization. But what I love about, you know, I think this case study here should really be published. So and the kind of approach they take down there in terms of that, if the Buckfast beekeepers won't sell Buckfast to a beginner in an area where they have native bees and vice versa. So they're aware. This is the beauty of actually sharing and communicating, is that if you're aware of what's in the area, then you work together to keep those things pure rather than you know, just doing it willy-nilly. So I think knowledge of what's in your area is really powerful. So the only thing I'd love to do more on Buckfast, because I think part of this could be that we haven't sequenced enough different things, and that although this is coming out like carnica, if we don't have other subspecies bees sampled, and in the data set it might superficially look like Carnica, but actually be a bit more complicated than that. It's just that we haven't sampled anything closer to it. So I do think we should include and broaden our reference data set. Okay, just to summarize a bit here. So basically Ireland's honeybees, the origins, one is that we have a native origin for Apis mellifera in Ireland, where it has colonized naturally after the last ice age, and they've been here about 8,000 years. Then we also have bees brought over from the UK over like 1,500 years ago, and bees have really infiltrated itself into all of archaeology, old laws, into our place names, into our language and everything because of this, these types, the bees being here for so long. And then we have non-native origins of bees, including Mellifera and Carnica, Ligustica, Buckfast and others that have been imported from various places within the last 100 years. So that's the status of what we have. The diversity is very good. AMM is widespread, but we still have it. We can see the impact of imported AMM, and we have good, distinctive and unique type of stuff going on in Ireland. Maybe Ligustica alleles are being swamped by Carnica mellifera in Buckfast, but this needs further attention. Nobody's really looking at this, and there hasn't really been enough Carnica, certainly in Ireland sample, to really investigate what's going on with the relationship between these subspecies. We've a survey just finished that we're about to now write up and look at the data, and we will present it back to all of the beekeeping associations in due course. But this just shows you, I'm aware of your the newsletter before Christmas, where your members have suggested about 80% or 85% of your members seem to have, I think they have AMM, and this is true in our survey. Out of, you know, here we have 324 answered this question. 90% of those who answered reckon they have native bees. So why this is important really to know what's what and where it is that, you know, I suppose everyone thought that maybe it was less than that the number of beekeepers having native bees was less. But if you have a situation, this paper, Snyder et al. Suggests that because the races belong to different genetic lineages, adapted to different environments, when you get hybridization between different subspecies, it disrupts this co-adapted gene complexes. They're too different genetically, and it results in reduced developmental stability and other issues, other traits that we don't particularly like. And then there's repeated observations that hybrid colonies tend to disappear over time unless managed by humans. And I do, you know, maybe we're seeing some of that in Ireland, whereby Apis mellifera seems to be pretty robust, but I'm now starting to look at monitor survival of hybrid colonies in the wild, and we'll be able to come back and report on that in due course as well. So what can we do? One is check what kind of bees you have and what bees are kept by others in your local association. I think, you know, everybody's entitled to have what bees they like legally. And people are very fond of their bees and, you know, shouldn't be chastised for what they have, because legally everyone's allowed to have what they have. But if we communicate more positively and we know what's what and where they are, then you can manage the situation much better. Where possible is maintain locally adapted bees, and in some places Buckfast or whatever it is have adapted to local, or they've been in an area for a long time and maybe doing well, and if they can be bred here and they're doing well, you know, you have to look at that. Support each other then at a local level to breed from and share good local strains, making sure maybe that there's enough nooks of local bees available for beekeepers, so via breeders and the association apiaries and local cooperative behaviours. And then if you wish to import bees, do so very responsibly, because these bees can disrupt thousands of years of adaptation and introduce more pests and diseases to Ireland. And this latter one, I think, has everybody very worried, and more so even in some cases in the hybridization. But Varroa came from imported bees, and you really want to protect what we have. What we can do as researchers, and what beekeeping groups and government agencies could perhaps do, is establish a genotyping service support. I'm looking into developing a non-profit company with Gabrielle and others to provide this and other services, but I will see. Work together to investigate the degree of impact of non-native and hybrids on native honeybees. So we have a responsibility to do that. Establish a management strategy. Now I've written up a draft management strategy and I've shared it with the associations, and I suppose the people in charge, we might talk about whether we publish these and get feedback from beekeepers to write the next draft, because it should be a live document that we then use as a tool to, I suppose, think about the future. And then improve again the provision of NUIGs for beginners and inform beginners and each other about really about the kind of stuff that's in here. What are the bees that are present? What do you have? Where did you get it from? The environment in one part of the country or in one type of environment is very different and might have a big impact. So where you get your bees can have an impact about how well they do. Okay, finally, I'm finished. I'm sorry, it's a bit long, wasn't it? And these are, sorry. These are some of the references that were in that talk. And my plan, as I said, is to, I've divided that into 3, and I plan to record it as 3 different talks and make it available for beekeepers if they're so interested. And right now I'll take questions. I'm going to stop sharing.
Wow, what can I say, Grace? You make a very otherwise heavy duty topic so easily understood. You have a gift with imparting knowledge, so thank you so much.
Yeah, I hope it was fun. I hope I didn't go like the clappers of hell too fast, but
no, there's compliments coming in there from some of the other participants, so thanks again. You made a couple of issues, or mentioned a couple of things there, the difficulties facing honeybees now at the moment, and I just wanted to share with those of you present that some of you would have enjoyed the webinar with John from Jersey. They've been tracking the Asian hornet and showed how one Asian hornet queen came into France and is literally responsible for the widespread colonization of the hornets there. And what he actually told us recently was that with Brexit and the increased ferry links coming from France, that we're more at risk now from the Asian hornet. And I'm just wondering, do you have any thoughts on that? And, obviously we need to connect up more and communicate better, but this is a very real threat, especially now. And I'm wondering, do you have any ideas or thoughts on that and how local bees might survive better against this species?
To be honest, I haven't, because I have been leaving that to others because I haven't had time really to think about that as well. It's a huge concern. I did see some, and I was probably shared again in one of the beekeeper Facebook pages. So I think one of you will have a better answer to that, of perhaps some defense. I mean, sometimes aggressive, docile bees aren't always the best in the world either, would be my take. I like my bees to have not too much attitude, not to chase me out of the field, but I'm willing to put up with some level. I don't know. So I think maybe open it to the floor. I would claim ignorance. I'm like a normal beekeeper in that context.
Yeah, I did actually hear as well last week that one of the bee inspectors in the UK, Megan Seymour, the Asian hornet nests they're finding in England are much smaller than they are in France. So they're actually thinking that maybe the weather isn't suiting them. So if that's the case, it would actually be very helpful for us. Yeah. But then if they adapt, if they do get in here and adapt. But look, you've gotten lots of compliments on the chat line there. There's one here. Thank you for a super talk. For a beginner, it was fascinating. Regards. And that's from an anonymous attendee. Any more questions coming in there? People, I think you've answered everything as you've gone.
Well, I try to make it understandable because I know how, I know how kind of inaccessible it can be, but I am planning on putting one together on genetics, the honeybee genetics, because that is, I think, very difficult as well. So I'm kind of planning to put a series of these together and making them available. I was looking at a YouTube channel, but yeah, I'm going to record this as 3, and then I've one on wild bee, or on the resilient bees as well, that I'd like to make available so that people understand it, maybe.
Brilliant, yeah. Somebody had a question there. It's just after disappearing. Is it possible to buy native bees that are resilient? You probably know more about that, Grace, and the work that's going on around the country.
Now, yeah, I mean, at the moment, people aren't advertising them as Varroa resistant. I guess we just carried out a survey, which some of you probably have done. So we've had about just under, I suppose, 350 responses, which actually is a little bit disappointing, but there are quite a lot of beekeepers who are not treating, and you know, a lot of beekeepers are hobbyists, they're doing their own thing. There are probably not enough beekeepers, I think, providing bees for other beekeepers. So we are starting a program now. I'm just starting very small because it's very hard to do, whereby we are systematically doing the tests for Varroa, and you know, the pinprick test, and recapping, and doing genome sequencing, and looking at differences between treated and untreated bees. And we plan to extend our methodology and make it available and try and build it organically from the ground. Because I think in Ireland, if we share this, and if we can get funding eventually, that we can provide support for beekeepers to record this and then feed in to a central database and have a kind of a cooperative, I guess. So when we get that data back, we do know that we have beekeepers who aren't treating for X amount of time, and then others for less time. And then some of the beekeepers have a lot of colonies, some have fewer. So I think we're going to put together a network of these beekeepers and see how we move this forward. So I guess we'll have a gold, silver and bronze standard maybe, and some of the gold people who have lots of bees and they're not treating and have lots of experience, find some ways of transferring their knowledge to everybody else and making bees like that available to everybody else. So most beekeepers think that this kind of research is really important, are willing to participate. So if we can get a breeding program up, but it's just very difficult in Ireland because beekeeping is difficult, cooperation is difficult, there's no funding, there's no real infrastructure. So we have a long way to go. But if we can, we are generating, we have now 26 genomes from drones, half treated and half untreated, and we're now processing colonies of treated and untreated and more beekeepers. So actually John McMullen's bees and Finglas Association are not treating, and like Pauline Walsh, there's other people who aren't treating, and we will get more people involved in the program and look at the genomes and look at differences and get all of the hardcore beekeeping stuff done, and then they can actually advertise their bees as being resistant. But it's very hard work.
Yes, but if we communicate and collaborate together, we can all help each other. Pat here has a very interesting question. I'd call it a heavy duty question. You have identified a unique genotype for the Irish Apis mellifera. Do you know how this is expressed in the phenotype?
No, and this is like this. I only discovered this at the weekend. I stayed up till 3 o'clock on Saturday morning processing this data, and so I need to look the next step, and this is stuff my 4th year students have generated. So the next step next week is to group those bees together, the bees that are green and see if there's anything. It's not, I wouldn't say they're not all the same genotype. They're all very similar to each other. I have to make sense of it. I don't know at the moment what it means. It means that I'm going to have to do some fancy analysis to see if I can find out if they're kind of ancestral, if they're old traits. So if these are traits that have left over from the bees from thousands of years ago that are distinct from more modern bees in Europe, or is it just that we've got a nice diverse population with lots of things? We're also sequencing genomes at the colony level at the moment. So we have 25 colonies to come back, and we're about to do another 45 colonies. And so I'm hoping that we will be able to detect that at the genome level. If we analyze the genomes of all of these colonies, that we'll see distinct genomes. So we're doing morphometrics as well, and that's a bloody nightmare. The foragers have been tearing their hair out, and I don't know how beekeepers have been doing it because you need very special, the scanner was driving us mad. So I'm trying to develop a method that we can all use, that you guys can use as well. And I will come back to you with that question. I'm looking at color. I'm looking at some of these colonies have very small bees and very big bees. I'm looking at the hairs. I'm looking at all the morphometric traits and color. And then the other thing I was going to say was, it's gone out of my head completely. Doesn't matter. What was I going to say? Oh. Yeah, so I will present all this data. We will write an article and provide this data as soon as we can. But we have to put it all together and try and make sense of it.
That's brilliant, Grace. As I said, the compliments are still flying in, and it's, as you say, a work in progress. So I think beekeepers in Ireland are very lucky to have people like you there with an interest in this topic and stuff. And
well, it's like CSI for me. Like, I have all this data, and it's like fingerprinting, and it's like going to see. For me, it's detective work and looking at patterns. I love jigsaw puzzles. Like, so this for me is like a big jigsaw puzzle. So I'd like to see, really what it means. Oh, the other thing I was going to say was, I've been in touch with the Natural History Museum in Dublin, and we have found bees. They found for me. They've ransacked their drawers, found bees going back to 1880. And the early 1900s, even some Ligustica from those original imports. So we're going to sequence the genomes of those bees. As soon as COVID lets up, we're going to go up there and beg them and sequence the genomes from as many as we can. And that will help us see if some of these green, that green cluster are like those old Irish ones, or if they're very different. So it'll help look at from a historical perspective, what was here then and what's here now. So that's going to be really exciting.
Yeah. We have Felix here, and this will probably be our last question. Are there any specific genetic phenotypic, my screen has been interrupted here.
Felix that came out of NUIG a couple of years ago.
I don't know, he hasn't said that yet to me. He's asking, are there any specific genetic phenotypic differences you have identified between Irish Mellifera and their European counterparts?
Hey,
it's Felix. Me, and by the way, he says yes, it's him.
There's only one Felix, right? Is an unusual name. How are you? We haven't looked yet. So we looked at, we got some Dutch bees from Marlene actually a couple of years ago, never got to process them. So I just did them now to see if we could dig deeper at this. Apparent, the mitochondrial DNA suggested that Irish bees, over 50% of them were identical to Dutch bees. So we wanted to look at that, see, look at that relationship a bit more. And they all seemed morphometrically or whatever. They had done the morphometrics and they seemed to be AMM, but I think we found one Buckfast in there. So somebody, I think it was Jonathan Getty, suggested that maybe the selection from Irish beekeepers has actually masked some of the signal. I have to think this through a little bit, but some of the bees that I thought might be Carnica were a little bit brown are actually seem to be pure AMM. So I'm really trying to get at this phenotype, and we really need, I think, to include more European bees and other these subspecies present in Ireland, really look at them to understand that better. So I plan to do some histology on the wing muscles some year with some final year project students, is actually to dissect the bees, dissect out the wing muscles and see if there's any difference. But I need to get, your question is really good, because we need to compare them now with European populations. I'm surprised at how little work has been done actually.
Okay, folks. It sounds like very interesting times ahead. The compliments are still flying in, Grace, there. We try and arrange, Stephen might be able to copy them and send them on to you. And we're looking for... there
about archaeology. I've been looking for archaeological remains in Ireland. There are some in England. Unfortunately, the archaeologist, it's just very sad, the archaeologist that I was chasing down, I found out that she'd passed away only in her 30s with cancer. So I kind of was shocked and saddened, so I dropped it completely. And I just yesterday had a conversation with somebody whose sister's an archaeologist, and so she's going to try and kind of chase up that again, because you're absolutely right, to get archaeological remains would be awesome. Yeah,
brilliant.
And I've asked a friend of mine, an ex-PhD student of mine who's in the Smithsonian, because sometimes the Smithsonian in America has loads of stuff from Ireland and England, so I've asked her to see it as well, see if there's anything.
Where do you find the time and the energy, Grace?
Thankfully, the beauty is, and this is really important part of the work of the university. This is, I can make time for this. I'm really lucky that in my job, I'm blessed that my job allows me to follow my own. Now I'm here at 9 o'clock at night and up at 3 o'clock in the morning because it's a passion as well. But I'm really lucky that I can. And actually beekeepers are lucky that the university is willing to allow us to do this kind of stuff, you know.
Yeah. This is great. Well, look, we'll help you all we can with samples and stuff, and everybody will keep members informed as you inform us. And there's a lot we can do. We can look at the grooming characteristics of our colonies, and we can look at hygienic bees, and we can learn more about this, and you know, we can all work together.
There's one thing that I should announce, and I haven't announced anywhere yet, is that I want to take part in a global project under COLOS. So the COLOS Survivors Task Force, we're starting we had a workshop just before Christmas, and we're going to do a recapping experiment. And so anybody who's interested, who has say 5 colonies that haven't been treated for 5 years, or if you're in a little group and you've got 1 colony but there's 5 of you nearby or whatever, if we get 5 colonies that haven't been treated for 5 years, that we would look at the recapping in those and contribute that data to we'll do 2 things. One is we'll contribute the data to the global project, but also we will sequence the genomes of those colonies that are put forward for this recapping, and look then basically at those who look at we'll be looking at the amount of ROA, the amount of recapping in the colonies that haven't been treated for 5 years, and then whether there's evidence of suppression of mite reproduction. So we basically will be uncapping 300 cells from a frame from each of those, just twice in the year, and counting the numbers of ROA and the different stages of ROA in any of the cells, just 300 cells. Now we're happy to do that in the university, but actually if a beekeeper is interested in doing that work themselves as well, they're welcome to do that. So just let me know, contact me if you're interested in being involved. You'll be very welcome.
Brilliant, that's great to hear. Grace, I'll be in touch with you myself tomorrow. What can I say? I'm down now here at the moment in the sunny southwest. My first time ever saying this, because the sun has been shining here all day.
It's nice today as well, I have to say.
Yeah, but it's been shining here all day in the kingdom, and my beloved husband tells me that it's pouring rain in the sunny southeast. So I'm enjoying my time in Kerry. For those of you still with us, we have our next webinar on the 24th of February, and we have Julia Jones from UCD. She's heading up the National Apiculture Programme, and she'll be there for us then. And I'm sure Grace will have many more webinars with you. You're very entertaining and a pleasure to listen to. So many thanks for doing this for us.
Thank you very much, guys. It was a pleasure, and hopefully we'll do more of them.
Okay, listen, thanks very much, everybody.
Thanks a lot. Time for tea.
Bye. Yes, enjoy. Bye-bye.
Bye.
Bye.